Hello everyone, my name is Michael Fiala, and I am a first-year undergraduate participating in the Undergraduate Research Apprenticeship Program, also known as RAP. This project on monitoring revegetation with NDVI and associated soil composition and microbial biomass inactivity was funded by the National Science Foundation EPSCoR project, improving water management, treatment, and recovery in oil and gas production. Transportation of a liquid or gas through pipelines has proven beneficial in providing fuels and resources over long distances and through landforms in a reliable and environmentally friendly manner. Although oil and natural gas production are essential to maintaining a modern society, these activities can also cause severe degradation to natural ecosystems. Reclamation of disturbed lands after pipeline construction is now required, and companies must show evidence of revegetation. One example of how revegetation efforts are monitored is via drones. Drones are autonomous aircraft that collect high-resolution images used to calculate the Normalized Difference Vegetation Index, or NDVI, to estimate the health of green vegetation. NDVI is often used in precision agriculture by observing the distinct wavelengths of visible and near-infrared sunlight reflected by vegetation. Healthy vegetation growth is then associated when chlorophyll found in vegetation absorbs visible blue and red light to fuel photosynthesis, whereas the cell structure strongly reflects near-infrared light. Therefore, monitoring revegetation using high-resolution imaging has proven successful, and linking positive NDVI values with soil health metrics could help connect above-ground recovery with below-ground processes. Soil microorganisms affect vegetation growth by influencing soil chemistry and structure and are both components and producers of soil organic carbon, which in abundance also improves soil fertility. Soil structure allows root distribution and the ability to take up water and nutrients, and microbial activity decomposes organic matter, cycling nutrients, and fertilizes the soil to develop a healthy soil structure. So the research question we asked was, could linking positive NDVI values with soil health metrics help connect above-ground recovery with below-ground processes for managers better to assess ecosystems' long-term revegetation efforts along pipelines? To investigate this question, we analyzed revegetation of disturbed lands after pipeline construction by using drone NDVI to determine high and low value sites. We then constructed soil plots and collected soil cores from 4 high and 4 low NDVI sites. After collection, we examined 4 soil quality indicators, including soil moisture, pH levels, soil water holding capacity, and organic matter, in addition to microbial biomass and activity. NDVI calculations and soil samples were collected in a revegetated area along a previously constructed 24-inch gas pipeline in Harrison County, West Virginia. 8 2-meter by 2-meter perimeter plots were made, each calculated by the NDVI values. Each pixel in the NDVI map is calculated in ranges from a -1 to +1. A 0 means no vegetation, and a +1 indicates the highest possible density of vegetation. High-value plots collected held an NDVI range of 0.38 through 0.55, and low-value plots had an NDVI range of 0.18 through 0.29. The soil quality indicators measured in microbial biomass identified no statistically significant difference between high and low NDVI plots. However, we could see a correlation between higher water holding capacity and low value sites and a difference between soil respiration in those high and low NDVI plots. Because both high and low value sites were collected from the same area located in a previously disturbed gas pipeline that has been revegetated, results suggest a connection between NDVI and soil health. Further research is required by including additional sites and soil quality indicator measurements to discover and figure out what is driving the differences in the water holding capacity and soil respiration between high and low NDVI values. Thank you for attending my presentation. I would like to thank my mentors, Dr. Paul Kinder, Dr. Ember Morrissey, Gregory Martin, Walter Vesca, and a special thanks to Lucas Kinder.